Hi, today we're going to talk about DNA. DNA is a molecule that stores genetic information which helps an organism grow, function, and reproduce. This is our DNA model, and I'm going to use this to explain the DNA structure. DNA has a double helix structure and is made out of 2 strands of polynucleotide. That means that each strand is made out of many nucleotides. So if we untwist the model and focus on one strand, it looks like this. This model isn't really precise about the structure of each nucleotide. So let's take this red part, which is a nucleotide, from the model and enlarge it to see what it looks like. So this is a nucleotide, and this is a phosphate group, and this is a sugar. So if many nucleotides stacked up together, the phosphate and sugar will form a sugar-phosphate backbone, which is this white part from our model. And this part is the nitrogenous base. And there are 4 bases in the DNA, and they are adenine, thymine, cytosine, and guanine. And as you can see here, they are paired up together, but they are actually not paired up in a random sequence. A is always paired up with T, and C is always paired up with G. That is because T and C are called pyrimidines, and they have only one ring. And A and G are called purines, which have double rings, and purines must pair up with pyrimidines because of the length. So if they're paired up together, they will have the same length. So the DNA won't have different length. Yes, and one more thing. The bases, like nitrogenous bases, are attached by covalent bond. And you will need this information later on in the video. So that's it for the structure of the DNA.
I will be introducing DNA replication and how it works. DNA replication is the process in which DNA, which is double-stranded, makes a copy of itself and makes 2 identical DNA molecules. Replication is necessary for the 2 new cells of the daughter to have the same chromosomes, or DNA, like the parent cell, if a cell divides. The mechanism of replication depends on the fact that any strand of DNA will serve as the reproduction template. DNA replication starts in a place called the origin, where the double helix of DNA is unwinded by the helicase, which is an enzyme that unzips the DNA by breaking through the hydrogen bond. Before we move on to the next step, we have to first introduce primase, and it is an enzyme who tells the other enzyme, DNA polymerase, where to start working. Primase place RNA primers, which is kind of like a starting point for the DNA polymerase to build. The next step of the DNA replication process is that DNA polymerase starts building from the primer, by grabbing the ATCGs floating around and matching the right pairs. DNA polymerase also has an ability to double-check if they matched the right pair, so it almost never makes a mistake. DNA polymerase also only builds from 3' to 5', but if you don't remember what that is, just know that the DNA polymerase on the 2 strands build in different directions. The problem is that the helicase only unwinds in one direction. So the DNA polymerase will end up running out of DNA to keep matching pairs on. That means that the primase has to keep putting primaries behind where the DNA is building to let DNA polymerase keep on working. The problem is that DNA polymerase is not that smart, so it doesn't connect the separated strands together. This means that there will be a lot of gaps, called Okazaki fragments, on one of the strands, and the DNA won't work. This is when ligase, another enzyme, comes in and fixes those gaps, making them one line by gluing the fragments together. And that is the final step of DNA replication. So after this step you will get 2 identical DNAs.